mentioned, most drugs were developed using male animals. And because of that, it might not be surprising that women have more adverse drug reactions than men. There's also some evidence that by developing drugs in male animals, we might be missing opportunities to discover things that would work well in women. And actually, I have some research I did when I was a postdoc in the lab of Rita Valentino, where we looked at a stress neuropeptide called corticotrophin releasing factor, or CRF. CRF does a lot of different things in the brain. One of the things it does is it's released during stress, and it affects a brain region called the locus coeruleus, which is important for arousal. So, and that's obviously very important during stress because you want to be alert. so, however, if this system becomes dysregulated, it can lead to hyperarousal, which is a symptom of PTSD, and can contribute to some symptoms in certain people with depression, like concentration difficulties or sleep disturbances. And so we were really interested, and since PTSD and depression occur more often in women than in men, we were really curious about whether this CRF would affect this arousal circuit similarly. And we found that females actually were more sensitive to the effects of CRF in this arousal circuit. And what is relevant for drug development is we identified that that's because there are some differences in the way the receptor for CRF works in males and females. And so in male and female rats, the receptor tends to bind different signaling molecules, which could have different intracellular effects. So in females, it tends to bind more this GS protein and signal through pKa. Males, it tends to bind this beta-arrestin protein, which signals through other pathways. And so one thing is that CRF receptor antagonists that target this receptor have been an idea for treating depression and PTSD for a while, because it turns out that there's high levels of CRF in many patients with those disorders. And these drugs were developed in male animals and then tested in either mixed sexes, or even some studies were more in females, and they didn't really work. And it could be because the receptor is different. So, excluding females, they didn't know the receptor was different at the time because they never looked at females till we kind of came along, but this could be one of the reasons why it never really made it to market. Another aspect of this is that there are pharmaceutical companies that are trying to develop drugs called biased agonists that actually shift the signaling of receptors for many different types of receptors, so that they may, in some cases, signal more through the G protein pathways, or in other cases, signal more through beta-arrestin pathways, thinking that by adjusting the signaling, they might have some important therapeutic effects. So our research shows that by kind of studying females and finding that in a natural state where females are more sensitive, this receptor is binding different proteins, maybe it gives you an idea for a drug that if we could have a drug that kind of pushed it more through the more resilient beta-arrestin male type pathway, that might be a interesting kind of target for this receptor. And so another problem with excluding females from preclinical work is that you're only kind of getting ideas from one sex. And by comparing the sexes, you might get way more ideas for developing compounds.